Well, welcome everyone today. Thank you so much for joining us for our MiDOG webinar series. We're very excited to have today for you Dr. Richard Harvey, who will be presenting on the skin microbiome in dogs. We have published some of these studies together previously, and he will give us a nice overview of our findings. MiDOG is an animal diagnostic lab. We use DNA analysis to identify all pathogens in all kinds of samples, not only canine skin. And today, it's my honor to introduce Dr. Richard Harvey. He graduated from the University in Bristol in 1978. He worked initially in mixed practice and then moved to small animal practice. He obtained in the UK and European diplomas and a PhD from practice. By now, he has published over 30 peer-reviewed papers and has co-written 6 books, the most recent one being Ear and Nose and Throat Diseases of the Dog and Cat, with Dr. Gator Haas as a co-author. And he's now actually writing the 2nd edition, so we're very excited for that to come out soon. If you have any questions during the meeting that you would like to ask Dr. Harvey, please put them in the chat. We will go over them at the end. And in the chat is also where you will find your certificate for the CE for today's meeting. You can download the document right there, add your name, and that's it. Or there is a QR code too that you can scan to go to a website to download the CE from there. And with that, Dr. Harvey, please take it from here.
Okay, thank you very much, Janina. A pleasure to be with you. It's 10 o'clock at night here in the UK, so I'm speaking to people in various time zones in the US, I guess. We're going to be talking a little bit about some studies that we've done over the last, I think, 3 years, looking at the bacteria on the dog's skin. And the initial studies was done with Dr. David Duclos, but the final one was not, but he was certainly here in the beginning. And in fact, the initial studies were based on a paper of his where he looked at whether scraping the skin before taking a sample and submitting a sample had an impact on the yield of the sample. So the very first papers we did were looking at whether scraping the skin of normal dogs impacted on the yield of the sample or not. And we showed that in normal dogs, scraping the skin before sampling made little difference or no significant difference to the outcome. Okay, Kate, if we can have the first slide, please. Can we have the next? There we go. So traditionally, when we've looked at the bacteria and indeed the fungi on the dog's skin, it's been termed a healthy skin, and on the healthy skin we find commensals and transients. Commensals are deemed to be present all of the time or most of the time, and although fungi are listed, not many of the studies have actually looked at fungi, so there's a lot of work to be done on that aspect of it for a start. Dogs, as we know, interface on a regular basis with the wider environment, and in doing that they pick up transient organisms. These are not part of the normal flora. They can live on the skin for a variable length of time, sometimes for weeks, sometimes for a few hours. And this can create problems when you're trying to identify what is part of the normal bacteria and flora of the dog's skin, because how do you differentiate between a transient and a commensal? Over the years, it's been recognized that the normal bacteria on the skin, the normal flora, the biome, if you will, helps to prevent colonization by pathogenic organisms. The classic one would be Staph pseudointermedius, the primary pathogen of dog's skin. And it's thought that the normal flora does this in a number of ways. It occupies favorable niches on the skin. It may optimally utilize local nutrients if those are in poor supply, effectively starving out a potential pathogen, and they can actively produce antimicrobial metabolites which suppress the growth of a pathogen. We also know that there is crosstalk between metabolites of the commensal bacteria and the skin immune system. If we could have the next slide, please. No, that's one too many. There's one between. There's one with a picture of dog skin. Keep going then. We'll come back to it. Next, next, next. We're missing one. Go back up. It doesn't matter. Okay. You can go down one. One of the interesting things that's become apparent from all of the studies on the studies of the microbiome of healthy dogs and allergic dogs and between allergic dogs and then dogs who are in remission from their allergic signs, is that by and large the same bacteria are present all of the time. In other words, whether you've got an atopic dog or you've got a dog who's got seborrhea, although there will be different numbers of bacteria and perhaps the proportion of the different bacteria vary from dog to dog, but they're the same bacteria by and large. In other words, there isn't a primary pathogen that causes the problem. It's a bacteria, it's there all of the time. And the reason it can cause a problem is thought there's either an abrasion on the skin which lets the pathogen in, there's an underlying allergen that lets the pathogen bloom, and then we have a situation where we get overt disease. Next slide, please. Right. If you look at an individual dog and you take samples from various points, nose, ears, chin, axilla, groin, perineum, foot, as indeed Dr. Kuzco did in this seminal study, she showed that by and large, there were more differences between dogs than there were on the dog. In other words, the bacterial load on the groin and in the axilla and in the ear was more similar than the biome between two dogs. And when you think about it, that's probably not too much of a surprise. We've already mentioned the fact that the dog goes through the environment, gets transient bacteria on it. There are sweaty areas, there are drier areas, and the proportion of different bacteria varies a little bit, but not much in the normal dog. And this is the critical thing. If you look at the bar graph on the left-hand side of the slide, you can see that by and large, the color zones are pretty similar all across. And when you consider that some of that was on the pinna, some of that was the axilla and perineum, it's quite surprising that the same number of bacteria, the same proportion of bacteria are present all over the dog. This is pretty much in stark contrast to the human situation, where we recognize greasy areas and dry areas, which have vastly different bacterial and fungal biomes. And so there's a lot of variation in people between various sites. Interestingly, just as in the dog, this flora is very stable. And there's a few years ago, there was a huge study from China where they looked at the flora over 4 years and found it didn't significantly change. So these microbiomes are very stable. And the issue then, of course, arises, well, why do they change and what happens when they change? Next slide, please. If the same bacteria are present on a dog with atopic dermatitis and a normal dog, how do we know if a particular bacteria is a commensal? Is it part of the normal biome? Is it if we find it on a swab, is it a commensal or is it a transient? Because until we know what is, we can't work out what is wrong unless we know what is normal. Next slide, please. The most frequent way of taking a skin sample is with a swab, and most studies are not temporal, they're lateral. So what you would find, for example, is they'd swab 30 dogs or 60 dogs in the groin and in the axilla, and then submit the samples for analysis. So you have a 30 dog spectrum, but it's a horizontal spectrum. It doesn't tell you whether you've got a transient or not. The only way that you can tell whether you might have a commensal rather than a transient is by doing something called a temporal study. In other words, you sample on a regular basis over a predictable time course. Now, there are some things you have to take into account. The changing of the season, for example. In the UK, we generally have a fairly benign, non-changing season. It gets a bit warmer, it gets a bit drier, it gets a bit wetter, but we don't get the hugely hot summers with humidity. We don't get the hugely cold winters, but it's something that you have to take into account. We also have to take into account something called the cutoff point. In other words, if you're sampling 30 dogs over 6 weeks or 8 weeks or 20 weeks as we did, at what point do you decide you've got a commensal and at what point do you decide it's not? So, for example, we could have a 50% cutoff if we have a bacteria that appears 50% of the time, a prevalence of 50%. Does that mean that it's a better study than one that has 70% or 85%? Well, to a certain extent, the degree of cutoff that you opt for is at the discretion of you, the investigator. But one of the difficulties of saying, well, we want an 85% prevalence because anything less than 85% could be a transient. The problem with that is you might miss what are called low abundance keystone organisms. In other words, something that is very important to the organism's cutaneous biome, but is not found in all instances on every animal in every site. Conversely, if you take a 50% cutoff, you might let in, if you will, bacteria that are not commensals, but in fact are transients. And this is a big problem that you have with establishing a temporal study. Next slide, please. So we used a brass guide, effectively a number 9 door furniture, and because you could sterilize that, we used that as a guide, and we swabbed within that guide. So that gave us a known area and a known number of bacteria. And then what we found was that we could call the density up. In other words, we know that the density of the bacteria, as you can see there, is about 1.1 times 10 to the 5 per square centimeter. And fungi are very similar as well. We then performed with MiDOG a 5-month study looking at axilla and groin from 15 dogs. Now, because we know that from Cusco's study that by and large an individual dog has the same flora in the axilla and in the groin and elsewhere, it didn't really matter where we went. But because of the ethical permission that we were working in, we weren't allowed to clip the dogs. So we went for axilla and groin, and we sampled every dog once a month for 5 months. So we did 15 dogs. If we can have the next study, please. The next slide. And this is the results that we had on this temporal study. And these are the organisms that we identified on the basis of prevalence. So when you look at 59, that was 59 out of a potential 70. So we have all of the samples above 39 are within 70%. Now, this gets back to what we called our keystone or our key organism. If we look at Staph pseudointermedius, fourth from the bottom at 25%, 25% prevalence, most people would say that Staph pseudointermedius is a very important bacteria. It can be found on normal dogs. It's the prime pathogen in dogs with bacterial skin disease. But the fact that it's down at 25% means that it's not a member of the normal flora, although you can find it on lots of dogs. If you look a little bit further down, you'll see the bacteria. You'll see that Malassezia species come quite nicely within the 70%. So they're part of the normal flora. And typically you'd find those in various areas of the body, the ear, for example, the perineum, the groin. And then we found 2 fungi that also came in above 70%, Vishnia Cozyma victoriae and Cladosporium. What's interesting about Vishnia Cozyma victoriae is that it's related to asthma, insofar as in a study of the New York asthma houses, they found that the presence of Vishnia Cozyma victoriae was inversely related to the incidence of asthma. And that's interesting because that goes back to the studies called the hygiene hypothesis and the recognition that the presence of a dog in the house might help to reduce the incidence of asthma. And of course, if Vishnia victoriae is inversely related with the incidence of asthma, if it's a member of the normal biome on most dogs, then the fact that there's a pet in the house means that Vishnu Victoria is also in the house. And it may be that there's a relationship, an inverse relationship with asthma. It's not quite as simple as saying a dog in the house stops a child getting asthma, because it's related to, again, like a temporal study. Did they have a pet but don't anymore? Do they have a pet, or have they never have a pet? And most of the studies looking at the relationship between asthma and Vishnu Vicova, or asthma and pets, didn't ask that question. It was just, was there a dog at some point, without trying to recognize when or how? And it seems that if you do ask the question, was there a pet but isn't now? Is there a pet, and has there never been a pet? Then you come back to the conclusion again that there is a relationship, a beneficial relationship between having a pet and having a reduced incidence of asthma. And of course, the dog owning population would be very pleased indeed to hear that there's a good reason for having a dog in the house, over and above the emotional and the exercise related benefits. You've got a protective benefit as well. Next slide, please. So on the basis of this temporal study, we found that we could nominate 5 members of the normal flora as being part of the core flora. Coagulase negative staphylococci, which are Staphylococcus felis, for example, rather than coagulase negative staphylococcus as in pseudointermediates. So coagulase negative staph. Sphingomonas, which is a gram-negative rod, that has been found on human skin and dog skin. Malassezia varietals, you've heard of Malassezia. And then we have these 2 fungi, Vishnia Kazima victoria and Cladosporium varieties. These were found on over 70% of the samples from 15 dogs over 5 months. So on the basis of our 70% cutoff, these are part of the normal flora. And that's the first temporal study that's been able to nominate what are called members of the core flora. People have suggested up till now that there may be a core flora, but on the basis of the temporal study, this is the first one that's been done using this particular form of next generation analysis to try and nail down with confidence the species involved. Can we have the next slide, please? Now, if we know the normal composition of the flora, both in terms of the species and the density, what happens when there's what's called dysbiosis? Well, dysbiosis occurs when there's a change in the composition of the normal community. We've already identified some members of that. So then we have to think, well, if we've got a dysbiotic skin, what's going wrong? And you can see that you can either have a loss of resident organisms, you can have a relative expansion of potentially pathogenic organisms, Staph pseudointermedius, Malassezia, for example, or there's a reduction in diversity. And in atopic dogs, what you tend to find is that the normal organisms that you find all the time are present, but there is an overabundance of Staph pseudointermedius, sometimes Corynebacteria, and a reduction in the coagulase negative bacteria. In other words, there's a reduction in diversity and an absolute increase in potential pathogens. And that's what's termed dysbiosis. It occurs in every situation where there's, if you will, a normal flora and an interface with normality. So in the gut, in the urinogenital tract, in the respiratory tract, and in the skin. And we call the ear, and we'll include the ear in skin as part of skin, of course, as well. So that's what dysbiosis is. If we look at an example on the next slide, please. So in an atopic dog, what we find is a reduction in the number of coagulase negative staphylococci, an increase in the relative number of Staph pseudointermedius and perhaps Corynebacteria and Malassezia. Now, these studies were done before this temporal study and nomination of core flora became apparent. For example, one of the interesting things is on normal dogs, for example, we found Sphingomonas, a gram-negative rod, as a member of the normal flora. But what happens in an atopic dog, because in the groin and in the axilla, you find Staph pseudointermedius, but we don't know what happens to the Sphingomonas because that cannot be grown on culture very easily. And this is one of the difficulties that's thrown up using new generation technology, in that a large proportion of the species and genera found cannot be cultured, or at least not routinely cultured. And that's then causing a problem from here, because if we say, well, that we've found Syphingiomonas as part of the normal flora in the axilla and groin of a normal dog, what happens to it in the groin and the axilla of a dog with atopy? We don't know, because those studies haven't been really done. And certainly the concentration, or if you will, the density of the flora in an atopic dog hasn't been looked at. And that's where we think we need to look next. The jury is still out as to whether atopy, or the dysbiosis in atopy, is a manifestation of an atopic skin, or whether dysbiosis is part of the genesis of the atopic state. In other words, is the skin, we know that the skin of atopic dogs is slightly abnormal. It's got a deficiency in filaggrin, which is important in making a fully functioning stratum corneum. If you've got a differential, if you've got a reduction in filaggrin, might you have in effect a leaky skin, and might then you find that crosstalk or filtering through the skin of epidermal metabolites alters the skin to generate an atopic state. We don't know. It's future work. Next slide, please. Okay, here's an interesting one. Squamous cell carcinoma on the nasal planum of a dog is associated with a local dysbiosis. As with atopic dogs, there's a reduction in diversity and there's an increase in staph pseudointermedius. Might, therefore, the percolation of metabolites through the skin affect the immune system, such that, for example, a papillomavirus might be activated, or a papillomavirus might be allowed to grow because of changes? And may that be a component of squamous cell carcinoma? We don't know. But it's interesting that the similar sort of dysbiosis that we've identified in an atopic dog occurs on the skin and around the skin of the nasal planum nasale if there's a squamous cell carcinoma present. Now, we're not saying here that the normal flora or the abnormal flora causes skin cancer, but there's certainly room to wonder whether the crosstalk between the epidermis and the dermis has some sort of impact on on the generation of a cancer. You throw in actinic radiation. There's all sorts of things that can be going on. So it's interesting. Food for thought, and more studies are needed. Next slide, please. Okay, so in our studies, we've identified members of the normal canine cutaneous biome. We've started to explore the implications of that. And the bottom line, of course, is that further studies are indicated. What happens in an atopic dog? What happens to the biome? What do the Syphingiomonas do? Where do they go? What happens in a dog with squamous cell carcinoma? What happens in the ear of a dog? We know the normal. Now we need to understand what generates the abnormal. If you will, our studies so far with dysbiosis have been descriptive. We know what happens, but they're not mechanistic. We don't know why it happens or how it happens. So further studies are interested. Further studies are indicated. Next slide, please. These are the references that we've pulled up that are relevant to this talk, which you can find on the presentation if you want to go into them. And the next slide, please. As an investigator, I am enormously grateful for MiDOG. They provided the samples, the swabs for the sampling, they provided the transport cost, and they did the analysis. I couldn't have done the work without MiDOG. I'm very grateful. Thank you very much.
Thank you so much, Dr. Harvey. That was a wonderful presentation. And yeah, we do have a question from the audience. If anyone else has questions, please enter them now so we can go over them. So someone is asking, I'm curious if there are any fluctuations observed in the normal flora over time. In the summer, when dogs are probably interacting with the dirt and outdoors, and temperature variation is driving subtle differences in abundances.
Yes, I think we can say observationally that skin disease in dogs in very hot, humid climates tends to be more aggressive. The Pseudomonas, for example, you get complications associated with the environment from that point of view. You could certainly say that in hunting dogs and in dogs who are moving through the outer environment on a regular basis, they're going to get experienced exposure to damage. Didier Carlotti, who was a prominent French dermatologist, identified something called hunting dog pyoderma. And this was when hunting dogs went over Mediterranean-style vegetation, lots of dry thorns and dry vegetation, and got microtrauma and overt scratches on their skin with nice pustules appearing 48 hours later. Interestingly, hunting dog pyoderma is more or less self-limiting. As the skin responded or started to heal, the pyoderma or the manifestation pyoderma disappeared. So yes, you can get damage to the skin, which is transient, hopefully, and then reverts to normal. You would anticipate in different climates where there is a different environment that the dog would go out and get more. I mean, for example, a dog who goes through water comes out with a wet skin. If he spends a lot of time in water, there's surface maceration. Maybe he doesn't heal quite as quickly in a hot, humid climate, or doesn't dry out quite as much. The bacteria live around a little longer, the transients hang around a bit longer. So yes, very definitely, your local environment will have a big impact on what the dog's experiencing.
I'd be personally curious what made you think of the brass number 9 to use that as a marker. I mean, I had never heard of that in any clinical study. I mean, it makes sense once you think about it, but to even come up with that idea.
Yeah, I just came up with the idea of that, and I went through toy shops and everything looking for something that might be round or at least that you could put against the skin surface. So we looked at plastic toys, and then I was just buying some hardware in a shop. That's what I need. So I looked at, I could use a 6 or a 9, it didn't matter. But it was brass, so you can sterilize it over and over again, whereas plastic would denature a little bit. So yeah, serendipitous.
Okay, we have another question. A recent review on dogs with atopic dermatitis has argued there may be crosstalk between the microbiome of the skin with the nose and oral cavity. Do you agree that there may be an interplay between body sites that could be impacting dermatitis?
Yes, for definite. Dogs' noses are covered in plateaus and small grooves, and it's been observed that the nose of a dog collects environmental samples, whether it's from nuzzling up to another dog, perineum or groin, or whether it's from looking at a smell on the ground. And some of these surface chemicals, hormones, debris, whatever, filter through these little grooves into the filtrum between the nostrils and then to the vomeronasal system. So the nose, as part of the skin, is part of the wider sampling system in the dog. Conversely, if the dog licks its nose, you will find flora on the nose. I've been quite interested in looking at the normal flora of the dog nose, and there's 2 studies where the nose, planum nasale, was swabbed as part of the investigation. And we find 3 types of organisms. We find oral bacteria, not surprising because the dogs lick their nose. We find skin bacteria, not surprising because they groom. And we find environmental organisms on the nose because the nose is part of the investigative tool of the dog. So the normal flora on the nose will reflect what the dog's nose is doing. So yes, you could say certainly licking the skin is going to transfer oral bacteria to the skin, and conversely, bacteria on the skin will transverse to the nose and get into the mouth.
Yeah, the bacterial translocation, we see that on all different kinds of body areas, so that makes perfect sense, yeah.
For example, you can find strep canis on the dog's skin. Strep canis is an oral bacteria. You can find porphyromona on the dog's skin. That's an oral bacteria, but they're transients. They're not there all the time. So although they were present on our samples, they were way down in prevalence, about 25–30%, I think.
Mm-hmm. Oh, one more question. I've come across publications linking gut dysbiosis to atopic dermatitis. What are your thoughts on the relationship between the gut and atopic dermatitis?
We discussed this in the pre-talk discussion because Janina is a gut a gut biologist, and I'm an observer of skin disease. So for very definite, there is a relationship. Years and years and years ago, I was in first opinion practice, and I diagnosed atopy in a small dog. And the owner phoned up 3 weeks later and said, "You're not going to believe this. My wife started giving my dog yogurt and the skin disease is gone." And I thought, wow. But whether it was a whisk diagnosis or whatever, I don't know. But there is a relationship between gut flora and the immune system. And there's absolutely no reason why the gut flora shouldn't have an impact on the skin immune system. Whether the gut flora has an impact on the genesis of atopic dermatitis or the atopic state is a different matter altogether. Janina might know a little bit more about that than I do.
Yeah, we definitely can see strong impacts between diet and the skin microbiome. We actually recently started working with Hill's Pet Food on a couple of studies to see directly the impact of different diets on the skin microbiome. So hopefully we will have more results coming out there soon. So yeah, we are what we eat in many different ways. But with that, we don't have any other questions, and I know it's getting quite late over there in the UK. Thank you so much, Richard, for for joining us this evening, and thank you everyone for attending this webinar with us. You will find the CE certificate in the chat here. If you have any difficulties downloading it, please give our team a call. We'd be happy to help, and we will be having more webinars later this year, so we hope to see you there again. And with that, thank you everyone, and goodbye.
Thank you.